I would advise never to use a title so long that you actually have to pause to breathe while you're saying it. So a title could have been Multiphoton Imaging of Hippocampal Place Cell Network Activity and Mouse Models of Neurodegenerative Disease, which is still actually probably a bit too long. That's mostly what I'll be talking about. So the overall topic is sort of neurotechnologies for probing neural circuit function in mouse models of neurodegenerative disease, so particularly Alzheimer's is what we're looking at. The technologies are hopefully of wider use, so hence I use the wider term. I'll give you a bit of a primer on mouse models of Alzheimer's in particular in the first bit of the talk, because not all of this audience may be completely familiar with that. And then I'll go on to the tools that we've been sort of building up and using. And finally, so actually this bit I think is probably the only bit which is in common. I gave some of that last time, but it sort of fits in with the overall pipeline. So I'll go through a little bit on this sort of whole brain ex vivo two-photon tomography. And then finally, Carmel Howe, who's a postdoc at Imperial, was supposed to be speaking about light field imaging, and what I'm going to do is give. So she unfortunately couldn't make it in the end, but she's given me a sort of a reduced version of her slides. So I will attempt to do them justice with no particular promises. So I sort of run a joint research group with Amanda Faust, and Carmel's a postdoc of Amanda's. But we sort of collaborate on many things. So just to give you a brief overview first of the kind of stuff that we're doing. So this first slide is all of the things from sort of my end of the lab. And it really is sort of, operates through a sort of a spectrum from neuroscience to neurotechnology. We've worked on a number of different neuroscience topics over the years. Right now, there's sort of two things going on in the lab, so I'm just sort of highlighting them. So one is looking at the mouse early visual system, and particularly the dorsal lateral geniculate nucleus, which to me is a very interesting topic, in part because you can sort of, in a reasonably simple way, get at some sort of interesting circuit concepts, such as sort of feedback from cortex and sort of what's going on there. So we had a paper a couple of years ago on this, and we're sort of there's ongoing analysis and hopefully soon some more experiments where we're actually sort of going into this with back with optical techniques again. The bulk of the experimental work going on right now is what I'm going to be talking about today, which is actually on mouse hippocampus. So we're doing two-photon imaging in awake mice doing a task. So this one here is basically receptive. When we're looking at LGN, one of the nice things about the early visual system is you can you really have sort of high dimensional and precise control over stimuli. So we can map receptive fields of neurons quite precisely, and this is an example of 20 or so LGN receptive fields. So on the x-axis here is a sort of a time axis. These are sort of frames in time, and these are sort of different cells on this axis. So you can see we've got what we call sort of on center and off center cells, and you see this sort of center surround sort of Mexican hat organization. So it's quite a nice preparation for that. But a lot of the technology, so that was actually done electrophysiologically. We've done a lot of multiphoton imaging work as well. This is just a gratuitous, pretty picture where we were doing just with a very standard two-photon microscope imaging layer 5 pyramidal cells in sparsely labeled pyramidal cells in transgenic mouse. So these were just expressing GFP. What I did talk about last time was the robotic, two-photon targeted robotic patching. So I'm not going to cover that today. And the work is progressing in the meantime. And what we're doing is moving to multi-patching. So our aim is to do two-photon targeted patching from multiple cells in vivo simultaneously. Right now, we're actually sort of testing out some of the algorithms for that in vitro, and you can see this is the multi-patcher. It kind of looks quite like one of the Spencer's setups that you saw the other day, which is a great disappointment, because when you publish a paper on this and you get the media people coming around saying, can we see the robot? And photographers say, we want to take a photograph of the robot, and then you explain what the robot is, and they're a little less interested. later. And anyway, the other thing, the other aspect of technology is actually the data analysis technology. So we develop, we collect new types of data, as many people in this room do, and it actually sort of provokes the development of new methods for analyzing that data. And we've used, well, this is some examples I'll show a little bit later on sort of dimensionality reduction methods, and then information theory has been a sort of a common thing used in the lab as well. So that's sort of my end of the lab, and there's a sort of continuum in one of our labs with my stuff down at one end and Amanda's stuff down at the other end, and a bit of overlap. And Amanda is really the more seriously optical side of the lab. We've collaborated, for instance, on source-localized multiphoton microscopy, which I spoke about last time. And then, in particular, she's been doing quite a bit of work recently on light-field microscopy, which we'll see later. In addition, she's worked on holographic optogenetics, which I don't have a picture for there. Okay, so first to give you a brief primer, brief intro to neurodegenerative disease and why we might be interested in it. So neurodegenerative disease is an umbrella term. I mean, Alzheimer's disease is an example of it. So is Parkinson's disease, but so are sort of prion diseases, motor neuron disease, Huntington's disease, and many others. So some of these have cognitive aspects to them, and some of them have less cognitive aspects. Dementia is really sort of the set of disease, well, it relates to the set of diseases which do have these cognitive aspects. So it's sort of defined as any disease which causes a deterioration in memory and or thinking skills. And of dementias, Alzheimer's disease is the most common and causes about 60% of dementias. In addition, in order of commonality, you have sort of vascular dementia, Lewy body dementia, frontotemporal dementia, and mixed dementia, where you actually have sort of multiple of these sort of aspects of the various diseases showing together. So comorbidities. We've sort of specifically focused on Alzheimer's disease, and there are good reasons for that. So worldwide, there are about 44 million people who suffer from it. In the UK, where we work, about 520,000, about 5.5 million in the US. Now, obviously, many of these are elderly people, so about 96% of patients are 65 years of age or older. However, there is early onset Alzheimer's. So in the US, there's about 200,000 sort of early onset Alzheimer's. It does affect a wider range of people. It's the 6th leading cause of death. Okay, now in part you would say, well, actually that number isn't. So there are 5 diseases which are much more likely to kill you. But of course, the thing about Alzheimer's disease is that it's not going to do so, or certainly it's not going to do so quickly. You'll probably die with it rather than of it, but it may cause a reduction in your standard of life. And of course, it's got big sort of economic and social consequences as well. And in particular, people with Alzheimer's often need sort of care, etc. as well. However, the deaths are actually going up, whereas those from sort of heart disease are actually going down. So it's sort of going in the wrong direction. And as we're starting to cure some of these other conditions, obviously Alzheimer's is going to become more and more prominent. And it does have massive economic impact. So by economic impact, I believe it may be the top or close to the top in terms of the diseases. So the global cost is estimated to be $600 billion, so 1% of the world's gross domestic product. So it's a significant problem. So what causes it? Well, that's been the target, obviously, of much research and debate over the years. You may have heard of the Amyloid Hypothesis, which I guess is probably the sort of the leading hypothesis, although the details have perhaps changed a little bit over the years. So, I mean, the primary issue, so this amyloid beta protein is the basis of this amyloid hypothesis. So basically there's an increase, so in Alzheimer's disease there's an increase in production of amyloid beta. There's also a decrease in the degradation of it. So basically the sort of natural enzymatic degradation of amyloid beta, and so that seems to be not working as well. And in addition, it's actually cleared from the brain. In fact, so you can see we've got some amyloid here. It's typically, we believe, cleared along the basement membranes of blood vessels, or at least that's one of the prominent theories. And so in Alzheimer's disease, the clearance mechanisms are actually stopping working as well, working as well as they should do as well. The cause and effect of all of these things is kind of complicated. It's not so clear which one comes first. There's a sort of complicated sort of interaction in a sense. But just to bring your attention to these guys here, the oligomers. So these are sort of small, very small clusters of amyloid protein, and these are probably the primary culprit for most of the brain problems that we sort of think about in sort of damage to circuitry. So they impair synaptic function. They change neural activity. We'll see they can actually both increase and decrease it, in fact. And they release sort of neurotoxic mediators from glial cells as well. So it's not just about neurons. Glial cells are sort of involved in all this as well. So there's some relation to sort of glial operation here. Now, this is what you commonly sort of hear about, these amyloid plaques. So if you have a lot of these guys around, then they can cluster into these amyloid plaques. And perhaps in maybe some of the earlier versions of the amyloid hypothesis, it was thought that these were quite important. I would say, from my understanding, the current sort of view is that while you tend to get these when you've got a lot of these guys around, it's not that these are the main problem. And in fact, when people have designed antibody strategies to attack these sort of big clusters, the amyloid plaques, you can do that, get rid of amyloid plaques, and actually cause more problems with the circuit than you had without doing anything at all. So they may be, I mean, they're important, but they're possibly a sort of a symptom rather than whereas the big problem is up here. But then we have other things as well, these ApoE4, which impairs clearance, and tau. So the other main sort of line of work is on this tau protein. So it's normally found in axons, and what happens is it ends up getting localized into cell bodies and forming these neurofibrillary tangles. And basically, by the time a cell has got these neurofibrillary tangles in, it's not really working anymore. I mean, you can't get recordings, etc. So it's sort of very much downstream sort of aspect. But locked in this this sort of nasty feedback loop. So it's multifactorial, it's complex, and we don't fully understand the effects of it on operation or information processing in neural circuits, which I think is something it's important that we do understand. I mean, obviously the pharma industry has largely operated on a model of sort of trying things out, sort of high sort of throughput, sort of trying things out, testing in animal models, getting it through to sort of clinical trials, and I think there's now a sort of a consensus that we need to understand a bit better what's actually going on with some of these drugs and their effects. So one of the principal hypothesis which has come up in recent years is called the Aberrant Excitability hypothesis, which is Palop and Mook. Actually, the earlier paper in 2007 is probably the main paper. This is a nice review paper. And as I said, amyloid beta can both increase and decrease neural activity. We'll see, I think, on the next slide some nice examples of that. So I'll sort of summarize those together as Aberrant excitability. And you can have sort of compensatory mechanisms, because obviously there are, the brain, brain circuits have a lot of sort of balance of excitation inhibition mechanisms, etc., in there, which can be triggered as well on this. And so you end up with sort of network dysfunction. And, this was sort of, so in this original paper, they looked at it in a number of ways. They looked at sort of field potentials, so his sort of controls, and this is the a mouse model which has got a modified amyloid precursor protein. So you can see this sort of like epileptiform-like activity. So one of the things that people maybe hadn't been aware of originally was actually you do in Alzheimer's get sort of epileptiform-like activity. And actually that's quite obvious in some of the mouse models where, for instance, one of the mouse models we were using, in fact we had to stop using because we would lose too high a fraction of the mice due to epileptic seizures. So that obviously doesn't tend to occur in people, but still the aberrant excitability is there. And so they also used ARC, which is an immediate early gene, the expression of which relates to activity. And sort of they found, like in some of these mice, you had this is black because basically it's clearly been going sort of firing like crazy, and in some of these other mice it's the other way around, or some other areas of tissue the other way around. You've got less, so it should be like this level here of sort of black labeled cells here, and you've got this one there's less and this one there's more. So this sort of summarizes that you should have something like this, and instead you're getting these kind of these sort of peaks and troughs. Well, we'll see something like that in the next slide, actually. So now this has mostly been looked at in neocortex because it's been easier to get it there. Obviously, one of the main things we know about hippocampus is its involvement in formation of long-term memories and its importance in a variety of memory. So these are things which are obviously particularly noted in Alzheimer's patients, and of course you do find amyloid plaques appearing at relatively early stages of the disease in hippocampus. So this is a thioflavin label amyloid plaque here, and clearly one thing that's quite important to do is to look at what's actually the operation of what's the effect of these on operation of this sort of memory circuitry in the hippocampus, which links through to neocortex as well. You can't really look at it in isolation. So that's just something to keep in mind. We'll be going back there. But just looking at cortex, so what actually happens? So this is some work from Mark Busch, who did his PhD with Arthur Connerth in Germany, who's recently moved to UCL. So we're collaborating with Mark on setting some of this up. He's given us a lot of very, very useful advice. And he did this originally with AMaster loading of dyes into neocortex. So that's what this is here, and he's sort of shown. So these are calcium dyes. He's labeled a bit of frontal cortex. So you can see these sort of cells here, and we can looking at sort of calcium transients corresponding to action potential induced calcium transients. And this is sort of what you should be seeing. Okay, in this mouse model, so it's the APP23 cross PS45. So it's actually got a sort of a couple of aspects of human Alzheimer's disease sort of genetically sort of knocked in. What you tend to see is, so this is another cell here, and you'll see here like this cell is actually more silent, so it's not firing at all. This one is maybe doing something normal. The red one here is hyperactive, so that's sort of firing at a sort of higher rate. And then when they've sort of tabulated this overall, they found actually, well, okay, 50% of the cells are in this mouse at this stage of its lifespan were about normal, but about sort of 29% of them were silent and 21% of them were hyperactive. So you had this sort of mix of silence and hyperactivity, so hypo and hyperactivity. And that related to how far the cells were from plaques. So now, as we said, the amyloid plaque may not be in itself the main problem, but most likely around, in the areas where there's amyloid plaques, there's also a lot of these amyloid oligomers as well, which may be more directly related to the problem. So what they found here was, so you can see, so the hyperactive cells are actually quite close to the plaques, and then further away you've got an increased number of silent plaques. So there's this sort of spatial distribution. And in this particular mouse model, they could look at how these progressed over time. So the amyloid plaques basically really started to appear strongly at after 5 months or so. So 8 to 10 months, they're sort of substantial there, and really on the same time scale, but perhaps even preceding it? Well, I mean, in time with it here, you have the development of these sort of silent cells and hyperactive cells. So it does sort of correspond timescale-wise.
Can I ask a quick question?
Yep, sure.
Do you know whether they characterize what cell types these cells are?
Not in this study. There has been a little bit of work looking at breaking it down into sort of excitatory and inhibitory interneurons. I mean, I think there's a lot more of that needs to be done. Right? Certainly something that's on our radar. Definitely interneurons play a strong role, obviously, in this sort of balance of excitation and inhibition. And one thought is that they may be, disruption of interneurons may be particularly sort of sensitive to having these sort of widespread effects. But I think we don't really know yet. So okay, what can we do about it? So there are actually a very large number of types of Alzheimer therapies which have been proposed. I'm going to talk about two, one that didn't work, one that does. So antibody strategies I mentioned, immunotherapies, you can use antibodies to target amyloid beta. That's been done. It does decrease the presence of plaques, but this is a paper from Bush and Conant in 2015. They found that you could decrease the plaques through the antibodies and actually make it worse. Okay, so in this case, you've actually got sort of a hypersynchrony starting to occur. You've got rid of the plaques, but you have this real aberrant activity going on. So maybe just targeting the plaques is not the best way to go. There's another major approach, which is to try and interfere with the generation of this of the amyloid protein in the first place, and to try and sort of suppress the generation. So there are a number of places that you can target in that molecular pathway, and BASE is this enzyme is a one particular one that's being proposed. So this is what we've sort of started. We've got a project sort of working on. So this actually comes from Ulf Neumann, who works for Novartis, and he's provided us with the drug. And basically, the earlier version of the compound was called NB360, and it actually worked quite well. I may have a slide on it later. Now, in the meantime, then they developed CNP520, which is a sort of a later version of the compound. Basically, this one here affects both base 1 and base 2. Okay, it works, but it actually turns the mice, their hair goes grey. Okay, so I think it would do the same thing in humans, but so base 2 is involved in sort of pigmentation kind of things. So this one here, they made it specific to base 1, and it no longer has that effect. It may not be the biggest deal to have your hair go grey if you think you're going to have Alzheimer's, but anyway, for some of us we're already there. So this is sort of the general sort of idea how it works. You've got in a healthy adult, you've got sort of normal production of brain soluble amyloid beta, and it gets cleared. Pre-symptomatically, you might have sort of increased production, and the clearance might be decreased, and all the clearance might be decreased. So you've got the formation of some amyloid plaques. Now, at the later stages, then then this sort of goes into overdrive. You have more and more amyloid beta plaques, irreversible loss of synapses, cell death, etc. So that's when it all goes wrong. And this is just sort of the cartoon version of how this sort of treatment works. That in the end, you're basically sort of decreasing the production of amyloid beta, and basically you may still have some generation of amyloid plaques, but this sort of phase of it is reduced. So it works. Okay, so firstly, just looking at the presence of plaques. And this isn't our work, this is from Keskin. We are trying this at the moment. So after treatment you have a reduction in the number of plaques, but most importantly, how does it work on, how does it affect the physiology? And it actually has a positive effect. So as you can see in this example here, we've got this sort of color code here green are the normal cells, red are the hyperactive, blue are the silent. So we want lots of greens and not many reds and blues. And after treatment, basically that's effectively what you get. So it does work in mice, okay? That's the caveat always with this, in mice. Now, they did then go on to starting clinical trials, but unfortunately Novartis have now pulled this drug after we started talking to them. And they've pulled it because of failure of another company's BACE inhibitor drug in clinical trials. It wasn't quite the same drug, but as I understand it, they have a sort of a complex risk model for all of these drugs, and I think that piece of information sort of pushed it above their risk threshold, and so they've said, okay, they're absolutely ruthless. No, we're axing that now. So that's too bad, but I think it doesn't detract from the potential long-term prospects for this kind of strategy and the importance of actually understanding what it's doing. Now, the reason that drug failed is not because it didn't work in Alzheimer's patients, but because there was a mild cognitive decline found in some patients that didn't have Alzheimer's. Okay, so it's an important need to understand, even just in, and I think that hasn't been replicated in animal models yet, but I think it needs to be. So we need to understand, why does this actually cause this cognitive decline? So there's a lot of unknowns still here, and that really motivates my sort of idea here to develop a technology platform for characterizing mouse models of Alzheimer's disease and the therapeutic strategies as well, with a sort of a high content approach, I would say. That typically, historically in the pharma industry, they were mostly using fairly crude behavioral tests with also some sort of very simple, usually just sort of immunohistochemical analyses. And what we, of course, want to do is be able to look at functioning circuits and see what's happening to them. So what are the requirements for this kind of platform? We need, in my view, cellular resolution imaging of neural activity, including in the hippocampus, which has the inconvenient sort of thing of being deep, to be able to image plaque locations as well as imaging functional signals in cells, to be able to do this in an awake mouse while it's performing a memory task. Okay, so that's sort of fairly important, and I think that's the bit that hasn't been done very much yet. It hasn't been done in the Alzheimer context, in fact. So one of the things that we can do, as we'll see, is to be able to dissociate memory encoding and memory recall effects. Now, I don't know that they'll have a separate effect there, but I think it's actually sort of an important thing to look at. What is actually, when we have disturbed memory performance, is it that you just can't recall, or actually is the formation of memories itself damaged as well? Now, one of the things that we're doing here at the moment is basically cellular or sort of population readout of memory. And you'll see what I mean in a minute. So rather than ask the mouse, I want to ask the mouse's brain. Okay, and it's because I can only ask the mouse very simple questions, whereas I think I can get a lot more information out of, what mouse CA1 actually knows in a sense. That doesn't mean we shouldn't ask the mouse as well, and it might provide sort of important additional things here. So we're doing that at the moment, and I've got a couple of students who are sort of working on sort of adding behavioral, behavioral readout as well to this. And then finally, the quantitative analysis pipeline is very important. You sort of very quickly find out how much is involved in this, and thanks to the work of people like Andrea, it's the tools for this are sort of coming together. And one of the things I guess I'd like to see, I've got a slide on it later, is effectively a modular universe where we can all sort of plug and play the latest tools, and I think actually the structure they've put together helps that happen. That's sort of data processing, and but then data analysis I think is an important topic as well. So that's something that we're looking at. Now, this is just to sort of cite the rival strategy, which actually isn't such a bad approach, which is the single photon. We've sort of discussed it, I think, breakfast, and sort of this sort of single photon and two photon sort of aspects. So this is the, there are a number of such systems, but the Mark Schnitzer one is probably the most commonly used one, where you've got a sort of basically an implanted grin lens and a microendoscope of some kind, and doing single photon imaging with that. So you can implant that directly down to the hippocampus, and you can do that in a freely moving, or maybe freely moving is not the word, but a moving animal. And it turns out that's actually quite important, and that actually has some advantages over what we're doing. So the pros, you can get many cells. The con of that is that you're really relying on demixing algorithms to dissociate cells which are overlaid in depth. And we have been playing with some of this data. We'll see that in a second, and that is actually a major issue. I don't feel I can trust it as much as I can trust the two-photon stuff. I think that's fair to say, but it's not to say that it isn't very useful. What we're doing is we're actually doing an aspiration, so removing a larger bit of cortex, so a grin lens and a guide tube can be smaller, so maybe cause less damage, which is good, and it's single photon, so the single photon signal is large. Okay, so. But you've got this, the fundamental problem is really this sort of demixing, both in terms of demixing cells, but also there's the neuropil contamination as well, which maybe can be completely demixed, but it's not 100% clear. So we're actually collaborating with Richard Morris's group at Edinburgh on this in rats. So this is just sort of one slide to say we're not ignoring that approach. So this is actually in the context, so this is not specifically in the memory context, but it's in a social cognition task. We have rats exploring, and basically they meet other rats, and then we have different conditions there. We're imaging sort of large ensembles of cells while they do that, and we find some cells that especially cells which have sort of different behaviours here. So we are taking that approach, and it's nice that you can do it in a freely moving animal. We're not doing that for this particular task. Maybe ultimately we will move to something like that, but this is what we're doing at the moment, and the reason is because we wanted to get as good imaging as possible, and so we sort of went with this head fixed preparation, and we sort of worked everything else around that. So basically we have sort of standard Nikon 16x objective. We have an aspiration with basically a bit of cover glass at the bottom. So we basically aspirate down to the level of corpus callosum, and then we can see through that in mouse sort of nicely down to hippocampal area CA1. And so we've got this sort of permanently implanted window. We do a viral injection several weeks before the experiments start, and what we're doing in this data I'm showing you now is actually delivering a GCamp6 Ruby sort of co-expressed, and this was developed by Tobias Rosa of München, and the idea of the Ruby is to have something, a sort of a structural marker which is not activity dependent in there as well, in the same cells. Ideally you had to have it sort of nicely labeled in the same cells. Yep.
So what is your success? So I don't know if you quantified it. What's the success of having both expressed versus one expressed, the other one not, and each other?
We haven't quantified it. Yes, it's not even with this, like loading them into the same virus. It's not perfect. Somehow one does express more in some cells and more in the other. I think it's probably a lot better if you just mix two viruses. It's a lot better than if you mix two viruses, right? So we're not doing that. He's actually made this construct which actually puts both in.
Yeah, that's better, right?
It's better. It is better,
yeah,
but it's not perfect.
It's not perfect. So
no.
But it seems
you do see
very
some.
good,
it is quite good. I've obviously shown you a reasonably nice one, but you do sometimes see areas where there's more red or more green kind of thing.
Ruby is not nuclear; it's cytoplasmic.
This one is. It is cytoplasmic, but the green is but it's not sort of associated specifically with the membrane. So you tend to see a sort of green, the GCamp bit on the sort of sitting around it. What I would say for now, I think the trouble with this is like waiting for Tobias to make a JGCamp7 version of this, etc. is kind of like it may or may not ever happen. So I think we are going to go away from that strategy. I think what we found in the end is rather than it being of use for so we initially said, well, actually, this is better we can use a ratiometric signal, maybe slightly better for movement, artifact correction, etc. it doesn't make a lot of difference there, to be honest. I mean, you can find cases where it does, but on the whole, you have to look hard for them. However, where it really helps is sort of registration and correction for movement artifacts, etc. To have, because the trouble with the GCAM signal is, one instance is this set, one instance is that, etc. And of course, okay, you average over time, but then that doesn't help if you want to be doing sort of, correcting sort of movements, etc. So we find it's actually very good to have a red signal in, but it doesn't matter so much what it is. So for instance, we could just stick another virus in the mix, and it doesn't matter whether it's co-expressed, because we're not using it just to mark the cells. Originally, we thought we were going to use that to find the cells, but in the end, the green channel is more useful. The other thing is we've got another project, for instance, where we've got, we're going to be labeling fibers that are going in, and we could, in principle, just use the red channel there, and as long as we've got enough fibers in, that should give it, as long as there's a structural signal, right? It doesn't matter what it is. So that's sort of what we're doing. The other thing we can do, and this is just sort of showing an example movie. We're typically doing a 500 micron by 500 micron field of view now. What we found with a 500 micron field of view, we need to do non-rigid motion correction. When we had a smaller window, it was okay to get away with just purely sort of translational movement correction. And the norm core algorithm is what we're using there. It works quite nicely. Yeah.
So, question, what's the speed at which you're acquiring?
30 frames per second. Yeah. So it's a resonance scanning. So it's basically a scientific. It's something a bit like a scientific hyperscope. It's actually an earlier scientific microscope with various mongrel bits added on that roughly approximates what they're now selling as a hyperscope. So the other thing we do is so several weeks before we do the viral injection, then 1 week before the first imaging sessions, we actually start the behavioral training. We'll see that in a second. So it takes us a week to train the mice to run in this little environment, you'll see. Then on the first imaging day, we start mapping the brain areas. And we do a methoxy-04 injection and then take the laser down to 720 nanometers and basically map out where the plaques are. Then we can relate, we can go back, and I've got a student actually working now on sort of going through and finding for each cell, as a function of sort of radius away from that cell, how much amyloid plaque is there. It's not necessarily going to quantify the amyloid oligomers, but at least it will tell us about the plaques. So we've got all that information. So I said these animals are head fixed, but they're doing a task. How does that work? So this is something in collaboration with Leo Karug of Neurodegenerative, a Finnish company. There's a little, so they run on, effectively they run a little carbon fiber platform which is floating on an airbed. Okay, so this aluminium apparatus here has got a lot of little holes in. We hook up the air to it, and basically this carbon fiber platform sort of floats on that. So the mouse runs, but it's the platform that moves rather than the mouse. The mouse stays in the one place. And then this is just sort of the scope attached. So we've done sort of two types of environments. Well, there's several types of environments. Commonly, we just have the mice running around in a circle. Okay, we found that's the most useful thing. They're good at that, and we can map place fields quite nicely with it. If you compare, I haven't got the comparison here, but if you compare this one, so this is actually an open field, approximation of an open field. They can run anywhere. It tends to be a sort of a longer-tailed speed distribution than you get in a tethered mouse, which is what we've compared it with. Basically we have 2 sessions a day, so over a week or so, they basically get up to around 50% running time, which is basically how we decide they're sort of fit for imaging. We want to maximize the fraction of the session that they're actually running, because then we can basically get more trials and map place fields well. Now, and I don't think I've got results to show you on the open field. We're still sort of compiling the figures on that. But, okay, here's the bad thing. Now, because these mice are head fixed, you don't get the vestibular signals which are important in generation of place fields. Now, somehow, that doesn't matter in a 1D task, okay? You can learn place fields, and we get very good place fields in 1D. But for 2D, you do need. So we do get some place fields here, but they're very much impoverished compared to what you would get in a true exploration. This was previously noted for virtual reality by Mayank Mehta, and so there were arguments with this sort of people doing sort of virtual reality sort of place field mapping around that, and we find exactly the same thing as him, but in our environment. Now, what are the advantages? So why would you use our environment versus a pure virtual reality? So the real reason is because it's sort of multimodal. You get visuo-tactile stimuli, etc. So you would hope it's sort of somewhat more realistic experience for the mouse than just like as when I went into this, I was thinking, well, like a stimulus just coming on the screen, what is it really going to mean to the mouse? Maybe let's try and do something a bit more realistic. I think it's not so dissimilar to virtual reality in practice, though.
I think 1D virtual reality is totally accepted.
Yeah.
Everyone uses
yeah.
it. In 2D...
2D is, I agree. It's not. I've seen people doing it, but we would say it's not. It doesn't quite work. So, okay. So we can do other slightly more complicated things. We can have the mice sort of go back and forth. We can have Y mazes, etc. We can do what I call a Theta Maze, where basically it's sort of like you make a Y maze, but you basically have a corridor that goes down the middle. So they sort of come around here. They've got to choose to go left or right. So we can do sort of working memory type tasks with that. We're still trying to work out how to get a good controlled delay period in this, but we've got a plan on how to do it. But anyway, this is basically how it works. Now, as I've mentioned, we can separately look at memory recall and memory encoding. So what we do in this 1 week of training, we basically train the animals on 2 environments. We call them FAM1 and FAM2. So we just have different carbon fiber enclosures that they can go in and out of. So this is an example of one of them. And so to test, then when we're doing our imaging, to test memory recall, we can put them in, say, for 20 minutes exploring environment FAM1, and then for 20 minutes exploring environment FAM2. And so they're familiar with both of these, what their hippocampal sort of CE1 cells is. So they move into a new environment, the cells remap, okay? This is this place field remapping. It's a sort of widely known phenomenon that's quite possibly damaged in Alzheimer's disease. But this sort of tests that remapping as a sort of memory recall process. On the other hand, and we always use FAM1 as the sort of control, so the first 20 minutes are always in FAM1. If we go from FAM1 into a novel stimulus, then this is testing memory encoding. So we can then look at what's happening as they're laying down, the formation of the memory, spatial memory of this new enclosure. So we have just a sort of little aside. Of course, we've had to develop a lot of sort of tools for this. This is our sort of current version of the processing pipeline. There's 2 versions of the processing pipeline actually. So Anne, the postdoc who's been doing the experiments on this, so she's running in a MATLAB version, basically, what we're doing on that data, and then simultaneously, I've got a couple of people working on sort of the next generation version in Python, which is trying to pull these things in, do it properly, and particularly incorporating this neurodata without borders sort of format, and so which the Kaiman have done as well. So with that, we should now be in this sort of modular world where you can grab norm core and then for motion correction, and then you might be able to, say for Roi segmentation, you might sort of jump to our Able tool, or you might use the Kaiman one as well. Then having ROIs, you might, so there's a nice tool, FISA, that Natalie Rochefort's group developed, which does a sort of an improved neuropil decontamination sort of measurement, and you can sort of plug that in quite nicely now. So we've added sort of things into to use that with the NWB. So that's the kind of the general picture we're sort of looking at there. And yeah, so we have our own version of the ROI segmentation. Now, actually, the data in this one that Anne is doing is actually using Kaiman right now, because she couldn't quite get able to work in all of the examples as well.
This is two-photon though.
This is with 2-photon, yeah, the 2-photon version of. But we're now kind of trying to reproduce it with Able and kind of tweak Able to be robust enough to pull out the data. And I think it's a good idea for there to be a variety of such things that are, and there are different constraints that are sort of tested on different situations, etc. So this is sort of our version Able, which was published in eNeuro in 2017, and is actually quite a, we found, for instance, on some of the public data sets, it works quite well. It makes different assumptions about the dynamics of the calcium time series, so it's not very strongly driven by the need to have sort of classical calcium transient looking things. And for instance, you'll find some cells that have got sort of things like this, other cells which may have nothing in it. He pulls them out fine as long as there is a basically a sort of a local correlation that it something that he's got to sort of grab on with, which is different to the correlation of sort of surrounding neuropil signals, etc. And it actually works quite nicely with dense synchronous spiking, which is, for instance, where the ICA-based algorithms are going to completely fail, right? So this is an example from dentate gyrus electrically stimulated in a hippocampal slice. The dentate granule cells are quite small and closely packed, right? And it's able to pull out sort of quite nicely a lot of different these different cells. Okay, so back to the data. So one of the nice things about this is we can actually go back to the same cells day after day. Sort of. So here's just a very early example that we'd done of looking at the same cells over 3 days. I think I've got the one over 2 weeks in a minute. Yeah. So then, and we also we can do it over up to 2 weeks or so. And this just shows there is a little bit of a kind of sort of evolution of the pictures, of the images over time. This is red channel, I believe. So we have a red and a green channel to look at that with. And we use things like sort of blood vessel locations as well to sort of match this as a control. So okay, we have our sort of raw images. We do motion correction on them. We do Roi segmentation. We do calcium transient sort of time series extraction. And then at the moment what we're doing is we're using OASIS to pull out spike trains. Actually, I would actually say this is one step that I'm not completely happy with. It works well in some cases, but I think there are some cells for which it will do things like trying to find the wrong baseline, etc. So I think we can sort of work on improving this this aspect a bit more.
These
Yeah.
These are not spikes.
Yeah.
These are just
Well, I wouldn't call them spikes. Sorry. Yeah. Events, but yeah. No, sure.
kind of.
Yeah. I mean, I can call them events. So that's just a terminology issue. But in terms of the
I
event
also say
estimation,
spike,
yeah.
not spike.
Yeah, no, sure. But I would say for many cells it works well, but there are enough cells where I think we can show that it works, at least applied without cranking the handles on. I find that the students doing this will not necessarily crank the handles quite enough and just accept what's there. And I think we need to sort of work on improving that aspect personally. Okay, so then sort of looking at the data, okay, what does it look like? So here's just an example. So here's traces from a few of the cells. The position, so this is just in sort of position around the environment. So basically it goes around to 360 and then the phase resets. So that's why you see these sort of jumps down. Now, when we're doing the training, what we're doing is we're giving a reward at a random location, well, actually, and following through the imaging. So we found if we give a reward to encourage them to run, if we give them the reward at the same location on every cycle, hey presto, there's a large number of place cells there, right? So, or place cells, reward cells, whatever you want to call them. So that actually turned out to be inconvenient. So what we did is we rewarded a random location around the environment. One thing analysis I haven't done yet, but want to do, is actually do the reward triggered averaging as well, just to see if we still have some reward-related activity. And that's sort of in the data we can look at. So these are sort of just some examples from typical cells, the kind of, so these here, so again, this is position around the circumference of the track, and this is the first, so each of these trials, so this is basically mapped onto space, so we take a time series. So one of the things here, just to mention, we turn these into instantaneous events rather than just keeping a time series. And there is one important reason for that, and that's because we want to transform that over into space, okay? Now, for instance, let's say you had a short versus a long calcium transient, or even deconvolved calcium transient, but as long as it's continuous in time, if you then transform that over into space, and let's say the calcium transient is lasting a particular period of time, you transform it into space. In one case, the mouse was moving slowly. In the other case, the mouse was moving quickly. That would be a major confound, because you would basically distort your response, and the slow one, it just looked like it was really long. Okay, so hence it needs to be instantaneous. So we come up with sort of just basically something that effectively preserves. It does preserve amplitude, but it's just an instantaneous event in time. And then that's what we're plotting here. Each of these is one of these events, and these are just sort of normalized overall for this image. And then each of these is a lap around the loop here. And you can see for these examples, they line up quite nicely, sort of vertically. So these cells have got reasonably good 1D place fields here. One thing we'll see in a second, actually, you might notice this one. It sort of seems to angle back a little bit like that, and just maybe sort of pay attention to that. But if we just average down here, then that sort of, we would call it sort of the average place field of that cell. And then what we've done is, for each of these cells, we've plotted those average place fields down here, and then we've just ranked them according to the positions. This is a very standard thing to do in sort of these linear track. And this looks very similar to linear track things people get in actual physical linear tracks, in virtual reality, etc. So yeah, and we can characterize, for instance, we can use spatial information. This is actually an approximation to the mutual information called the Skaggs information rate. It actually, I don't like it. There's a problem with it I can maybe discuss with people later, but it's accepted as the standard thing in the hippocampal field. We can just as easily compute the full mutual information, which actually better captures so this thing doesn't know anything about variability across trials, okay? Whereas the full mutual information does. But the numbers in practice don't look it doesn't change things very much. So these numbers look sort of fairly sensible. So I mentioned this this sort of slope. It seems like this example here, not really on that one there, but on this one here, it just seems to be shifting back a little bit every trial. So do we see it? So this is, let me just sort of take you through this figure. This is all of the data, okay, from one mouse, okay? There are little lines along here which unfortunately are not visible on this. So you can see these sort of blocks that correspond to individual cells. So that's one of the blocks there, okay? That's another block there. That's another block there. And it's
one cell, 1 day.
Sorry?
One cell, 1 day.
This is just on 1 day in this case, yep. So this is 139 cells, one mouse, and then they're sorted by this Skaggs information. So this number. So for each of these, there's a Skaggs information value. So basically, these 5 are examples in that, right? So basically, the ones down here weren't place sensitive. The ones up here were. You can see in many of these, there is actually this backward shift. So these 3 examples here, quite prominent. It's not in all cells, though. And it's a different slope for different cells. So we're trying to figure out what's going on here. I mean, if it was just an artifact of, of course, it should be the same for all cells. So it's not that. So this is potentially interesting. Mehta reported something similar quite a few years ago, but in his case, they didn't shift back. To just grew back in a sense. They were still responsive at the initial location, but they just started firing earlier. Whereas in this case, they seem to shift back. So we're still puzzling a little bit about what this is and trying to sort of think of controls to do it, etc. on it. But we certainly do find some of these do. And one thing to say is, because if you think about how you should also calculate the information, the spatial information, you're just sort of assuming that a trial is a trial when you're doing that, right? Whereas actually, if it's shifting back, you probably should sort of factor that into your information estimation. So if you do that, and I have done that as well, you can sort of re-plot these, and it looks a little bit cleaner. But yeah, yeah.
Do you think it could have something to do with the mouse anticipating?
Yeah, well, maybe. Yeah, exactly. I mean, it's sort of learning. Yeah, exactly. What it knows that is coming. There are things we can look at that we haven't looked at yet, like, you know, if the mouse is running at a slower speed versus a faster speed, does it change how much it happens and things like that. So there's a few analyses we can do. We could also do things like making disks of different diameters, but we'd have to do new experiments for that. But so yeah, it's just something that sort of popped out that was quite interesting, that as far as I can see hasn't been observed before. And I wasn't expecting to see something like that in calcium imaging, really. The reason I looked for it is because a theorist, Tony Burkett, who was doing a sabbatical in my lab visiting from Australia, had basically asked us to look for that, because he had some ideas on how, using some sort of Hebbian mechanism, you would see something like the Maillard matter phenomenon. And so we went and looked, and lo and behold, we saw something. But yeah, so okay. Now of course, leaving that aside for now, the sort of thing we want to look at, of course, is remapping. So here, for instance, are all of the cells in grey are all of the cells for one region that were ever active, in the sense they ever fired one event or more. And there are I think there are 196 of these in this particular example. This is one mouse. No, sorry, this is two mice. This is an average over two mice here. We're adding more mice into this picture. And so you can see 164 of those had place fields to environment A, which is our FAM1 familiar environment. If you then take the mouse and put it in environment B, okay, then what you find, if you look at the cells which had significant place fields, it's now the blue ones. Okay, now you can look at the intersection of the blue and the red there, and that's these ones here. So there were some cells that had place fields in both environments, but there were many cells that had place fields in the one environment and not the other. And if what you do is you take the responses to environment B and resort them according to the order that was in this one here, you then get this scrambled thing here. Okay. So there are some of these cells are responsive, but you've had this remapping. Okay. Now, if you then instead take these same cells and resort them according to their place field preference in environment B, you also get this kind of sort of nice sort of diagonal map. So they do have place fields, but it's just that only some of those place fields are the same. Only some cells have place fields in both environments, and there are many cells, including cells that didn't respond here, that also have a place field in environment B. We should show I've asked my postdoc man to make the scramble version of this one as well. We should have 4 to sort of go back and forth, but that's the general sort of picture that shows. So it lets us look at this sort of remapping. Now I don't have an answer for you yet on, well, actually, in our Alzheimer mouse model, what's the effect on that? So we are looking at the 5xFAD mouse model. It's a fairly aggressive model of sort of early onset Alzheimer's disease, which shows amyloid plaques and shows behavioral effects, etc. And we're doing experiments at the moment. We've had, I think, three of the last batch of mice in that cohort were 5xFAD. So we're hopefully soon we'll sort of get an answer on that. Yep. Okay, so then what other analyses should we do from that? I just wanted to, maybe I can just do this in chalk just to show some of the population analyses. So I was showing you some things which were really single cell analyses of single cell properties. So the place field is a property of a single cell. But it would be interesting to know what is going on at network level as well. When we're looking at that, basically we've got a matrix, C by T. C is the number of cells, T is the number of time bins that we're looking at. There are a number of things that we can do with that. So I'm now going to sort of talk about dimensionality reduction methods. And you can use principal components analysis to do this, or multidimensional scaling, or various other methods. Our main favorite in the lab is multidimensional scaling for a number of reasons, but we do often use PCA as well and other methods. And most of what I'm going to say here applies really regardless of which you use. So you might do several sort of complementary analyses here. So let's say you took each, say two cells here, so I might call it this cell here, and took the vector over time, and this cell here, and took the vector over time there. You could compute the correlation coefficient between them, or actually what we tend to do is compute the mutual information between these two time series. And then you've got a matrix, C by C, each element of which has got the mutual information between these two time series. Actually, what we'll often do there is turn it into an information distance, which is sort of D equals, D equals 1 minus I over the joint entropy. So this turns the mutual information between these two into how far are they apart in information terms. So we then do dimensional reduction on that, okay? And we get, you might keep k components, sort of k dimensions, and we can actually look at sort of eigenvalues and eigenvectors and see how many we should keep, and sort of k by c. And then for each of these cells, we can sort of plot that. I'll plot it in sort of 3D here, and say we've got sort of this cell here, this cell here, this cell here, etc. In this space, that shows how far apart are these cells in the information that they represent. Okay, so this, it sort of brings in the single cell properties here, and but plots them in terms of information space. So that's one kind of analysis we sort of do on this. A second, which I'm actually going to show you some results on here, uses a different. So here we've had some kind of distance measure or dissimilarity measure. Instead, let's look at the distance between two time points. So here it was C1 and C2. Instead, let's look at T1 and T2. So what I'm looking at here is the vector of neural population activities from all of the cells at time 1 and at time 2. And so you've got a sort of a vector there. How different are those vectors? You can use something called the cosine distance. Cos theta equals, so we'll call that R at T1, R at T2, sort of dot product over R of T1, R of T2. Okay, so that's basically, if you think of these as vectors in high dimensional space, it's the angle between them, or the cos of the angle between them. So that's a cosine similarity, and then the cosine dissimilarity is 1 minus that. So let's do that, and then for all of these time points, and then we've got this matrix which is T by T. Now the catch with doing this is that T can sometimes be big, but let's say we do that, and we then do dimensionality reduction on this, and keep, again, k components. And then we've got this sort of k by t thing, and what we can plot in k dimensions basically is the sort of trajectory of the population activity, through this, through this space. Now the interesting thing here is, as long as we can put things together in time, we can compare them, and we can find what we're doing here with multidimensional scaling. Anyway, is we're finding basically a sort of projection which preserves the distance measure that we've actually sort of set here. So we've defined that cosine distance measure, and we're sort of finding the projection into this low dimensional space which preserves those distances. So we can actually see, or for instance, when the animal was in environment 1, maybe it's sitting over here, and then maybe it goes into environment 2, maybe it goes over into a different part of this, this space. So this is sort of becomes sort of quite a powerful way of looking at this. And so, this is, for those of you sort of familiar with the systems neuroscience area, the neural manifold analyses are a big topic right now. This is sort of our take on it, and it's something I was doing before you got called neural manifolds and the name became cool, but it's...
What's the characteristic time difference across which there is meaningful shared information for any two pair of neurons?
That depends.
Timescales?
Yeah, I mean that's something one has to discover, okay, for any data set. I mean, in our case, we typically have operated this at a sort of, I think it was 100 milliseconds or something like that time scale, and it seems to be fairly robust to the time scale at which we choose that, but it's that kind of order, I think. Yeah. Yep.
Have you tried other approaches that are a little bit more, let's say, that gives you a less linear representation of this data, like LDF?
So MDS tends not to give, MDS is a nonlinear method, and actually if you look, actually a nice thing to look at is the sklearn. There's a set of examples in there, and you can actually, I think it was sklearn, and you can show the projections out. The LDS stuff tends to not give a good, there's a nice example they've got that MDS sort of preserves the distances and it does a nonlinear dimensionality reduction much better than that.
I'm talking about LFATs.
Sorry?
Not LDS, LFATs. So basically you take your activity and you have basically a network that
I see.
compresses everything into a dynamical system that can reconstruct the spikes. And then you can use that little dynamical system.
Sure. We haven't looked at that particular method. I mean, there are many methods. My experience is they mostly tend to give very similar answers when we compare.
This one is that in some sense deal also with Nico raised, right? That you don't know the time scales at which these things...
One of the things that we've done to compare these methods is we've looked at so if you look at the eigenvalue, so out of all of these methods, you can get something like an eigenvalue. Now, it's probably sort of mathematically illegal to call it an eigenvalue except for PCA, but it actually does tell you sort of how much of the variance you're capturing in a sense. And so you do this and you get, say, this eigenvalue may capture this much of the variance, this one, maybe the rest of them in the noise here or something. So maybe there are two eigenvalues here that are sort of capturing what's going on. So what you might do is say, well, how many of these eigenvalues do we need to account for 90% of the variance or some arbitrary fraction of the variance? And then we'll call that D. And this was something that was done in Karen Harris's recent paper for PCA, and they called it the dimensionality. But you can do that for any of these methods. And then you can compare the dimension. For the different methods, you can compare the dimensionality versus the number of cells. And basically, you can look at this data, for instance, and see that MDS will need less dimensions than PCA for the same number of cells to capture the variance. So a nonlinear method can do a bit better than a linear method because it's got more to play with in a sense.
So the eigenvector air quotes that you get with this secondary method you introduced gives you a bunch of neurons that are mutually correlated and the associated typical time delay between them firing?
It gives you...
With the first method, I see how you would get neurons that fire together, typically grouped together.
Yeah, in this case, it's actually it's not telling you about the neurons, it's telling you about the patterns. Right. Okay, so the eigenvectors are actually the components of the patterns across time.
Okay.
So one of them might be something like the average.
So snippets of firing patterns for a specific group of neurons.
Yeah, exactly. So Okay, so anyway, that's some of the ideas we're playing around with, and you can. Now, the nice thing about this task is you can make it. So I actually use this in one of my teaching courses. You can make a nice little model which looks like this experiment. And where's my pointer? So I don't draw on something here. So this is basically a Poisson neuron simulation of exactly this task, and you can play with the noise levels and things like this, and do this exact same analysis. So this is this analysis applied here, and you find we're plotting it in 3 dimensions here, and you can basically you can recover this 2D trajectory. In this case here, you add enough noise and then you can't. So what does it look like for real data? Well, this is kind of early analysis. Somewhere, but it's a bit bodgy, but you can actually still kind of recover a distorted version of the analysis. Now this was with a relatively small number of neurons, and we can add more. In principle, we could even throw mice together and do this analysis. That's something we might think about as well. It's nice when you can do analyses where you can actually just sort of throw it all in and find a representation, sort of as if you had recorded from many thousands of cells in one mouse, whereas actually those cells are in different mice. Obviously, there are constraints on that. So just to sort of summarize this section here, we've developed a pipeline for imaging calcium signals in population of neurons during behavior. I mean pipeline not so much in the software sense, but in the whole thing. We applied it to spatial memory, and particularly a circular linear track and open field task. It's also possible to do working memory tasks with a Theta Maze version of this. And we can look at the contribution of neurons to behavior as a function of their individual amyloid load profile. Our aim here is to observe recall of old and formation of new spatial memories to track these neurons over weeks. We're testing this in the 5XFAD mouse with this NB360 drug at the moment. So the next thing that we're doing is part of this pipeline. So, okay, we do our in vivo experiment. At the end, we kill the mouse, take the brain out, and process it for serial 2-photon tomography. So this is a tissue site system, and basically image, say 100 microns thick section, tile. So you take the whole mouse brain, you've mounted it, you tile imaging sections, image all of those, slice that off, image the next lot, etc. And then these sort of slices float off in the bath. We found if you cool the bath, the slices sink to the bottom, which is much better. Otherwise, you get bits of sort of tissue will sort of float back over your sample and corrupt your imaging. Now, one of the things that you can do is actually to take these slices out and then do immunohistochemistry on them. And this is actually something that we had a group of undergraduate students looking at last year, and they got part way to sort of getting that going. So we have this sort of a multiple sort of steps in this kind of platform. Now this just shows one of our 5XFAD mice. This is an old 5XFAD mouse, so it's actually this expresses quite early. So this mouse's brain is a bit of a pincushion, basically. The purple magenta here is the methoxy label for amyloid, and the grey is the Allen basically autofluorescence, which we use to register with the Allen mouse brain database. So we can do this. We can transform all of our mice onto the Allen mouse coordinates to have standard brains that we can then do things across multiple animals. And then the other thing we can do is to trace connections. Now, our version of this tissue site, we've sort of got optimized for high resolution, and it takes up to 4 days or so to image a mouse brain. We can do it quicker if you want to do less resolution or less of the brain. Sainsbury Wellcome Centre have instead sort of modified theirs to image multiple samples per day, and they get lower resolution. But I'll show you why. I think I need to quit that and start preview. There it is. I'm going to drag it across. So, I mean, you can see that we actually get very fine. I mean, this is a transgenic mouse which has got a lot of label in. We're also doing this with viral injections where we've injected, for instance, rabies virus into a much smaller number of cells, and then you can really trace individual axons, at least for some distance. And we'll see an example of that in a minute. But you do get very fine sort of spatial resolution on this. So what we're doing is we're taking the in vivo brains. Now, we've obviously got this massive infarct, which makes it fairly easily to sort of match. But we've got people developing image processing algorithms to match exactly and transform into the LN mouse brains. We take our in vivo imaging, and then we can look at across the whole mouse brain, the location of amyloid, and potentially any other injections that we might make. Yep.
You were also looking at autofluorescence?
We use, so exactly, there's an autofluorescence channel which is used for the registration with the LN mouse brain. So it's just used for structural information. And this is a...
What kind of information is it telling you?
It gives us... maybe... Yeah, but basically, I mean, you get a kind of a sort of a grey image of basic sort of outline of structures, not anything that's high enough resolution to use for individual neuron level imaging. But now it's actually, having looked this up a bit, I mean, I think this autofluorescence is coming from 3-photon excitation of tryptophan. So there are people who've sort of looked into this. It's a sort of a standard signal that they're sort of using for this. Yeah. So it's what people are normally using to register with LN. Yeah.
Okay, but
Yeah.
you don't know where it's coming from?
Well, I mean, it's coming from tissue, but not specific to cell type or anything. I mean, we've thought about exploring that further and trying to look at, for instance, can you develop, Nissl state equivalent for autofluorescent signals, etc., like that. But we haven't taken that any further. And I think there is interesting things to do with that if you start to do a bit of spectroscopy with it.
Yeah.
But there's definitely it's a reasonably strong signal. Yeah. I mean, off the top of my head, I don't recall the exact wavelengths, but we've got them. Yeah. Okay, let me go back to the slides if I can actually find my way back. Okay, so yeah, and just to mention, we can actually do the tracing. So here's an example where, so this is actually a rabies virus injection into the lateral geniculate nucleus, and then that retrogradely infects cells in visual cortex, and they turned out to be both layer 5 and 6. Probably the layer 5 cells, because we've probably got some injection into the VLGN as well as the DLGN, the ventral LGN, which is basically another structure which has been of interest recently, but it has a slightly different projection pattern from cortex. So basically, we can trace them a certain distance, but we can't trace them all the way to the final destination, unfortunately. I mean, you could if you had a much smaller amount of cells labeled, but with the rabies virus technique, we're getting too many. But in principle, it sort of lets us do that. So basically, our idea here is that to locate the region of tissue in the in vivo imaging, register it with the LN mouse brain database, take slices out of the tissue site bath, then we know we can actually find, by a matching algorithm, find where they came from in the whole brain tomography image, but then do immunohistochemistry on them. So we can have sort of a pipeline all the way sort of going through to get immunohistochemistry from sort of selected regions. So we don't have results on that yet, but that's where we are in the process there. And in principle, then, if you've done that, you can do things like FISH as well to do sort of in situ genome analysis too. Okay. Now this is the end of the first talk. I've got another bit with Carmel's things. I don't know where we are time-wise. What time is it? Are we kind of?
10:50.
It's 10:50.
Yep.
So I've overrun the coffee break and eaten into Andrea's time. I mean, what do you want me to do?
We have
Yeah.
our
Yeah.
We have a lot of open time, so
Okay.
let's keep going.
Shall I just keep going? But do you want me to stop for a coffee break now, or someone's nodding? They need coffee.
No,
That's fine.
I was nodding to keep going.
Okay.
Just finish your presentation.
Yeah, we'll just keep going.
Okay. It probably won't be long. Feel free to grab a coffee while I'm talking.
Quick question for you. It's probably really quick to get through the airport.
Oh, yeah.
It's in Santa Barbara?
Yeah.
Yeah, 20 minutes.
Okay, cool.
But are you going to be checking any bags?
No.
Okay, yeah. So just 20 minutes.
Okay, great.
Okay, just to mention some of the people involved in this. In particular, Anne Go is a postdoc who did most of the in vivo data collection there and has really developed that whole platform. Jake Rogers was sort of mouse behavior expert. This is Carmel, who couldn't be here, but I'm going to talk about her work soon. And there were quite a few other people here as well. See how you might recognize since he visited Spencer's lab last summer. Okay, let me find Carmel's talk. Okay, so with Amanda and led by Carmel, who's a postdoc with Amanda, we were looking at light field microscopy. In part, we'd had an interest for a while in trying to do volumetric voltage imaging, and we tried various things to get good voltage imaging going, and it's a difficult problem, as I'm sure many of you know. But I mean, on the way to that, we ended up sort of somehow at this sort of light field microscopy area. Okay, the general idea to detect functional activity with fluorescent indicators. We can use calcium or voltage indicators. And what ideally we want is sort of high temporal resolution, 3D volume, single cell resolution, and light efficient. And obviously this is a bit of a fantasy list, but there's many people working towards it. And maybe light field microscopy might give us sort of one way of sort of doing this. And of course, one of the problems is that action potentials are short. Even if you're just looking at sort of postsynaptic potentials, you still only have sort of 10 to 20 milliseconds or so to count photons. I probably don't need to go through the optics stuff here, but we've got sort of a standard sort of wide field microscope. We've got a point in the sample space which becomes a magnified point on the center. With a light field microscope, you place a micro lens array at the native imaging plane of a 4F optical system. And then what this does is sort of multiplex 2D spatial and 2D directional information onto the same image sensor. So rather than just using sort of a point imaging, you're actually trying to sort of extract more information from the spatial information using this sort of micro lens array. So this sort of compares what you might see under wide field and light field conditions. So you've got this micro lens array here, and then the camera sensor that you're using to pick up the signals here. So if you're just doing this in wide field, you might see something like this, the equivalent picture in light field. So basically you get effectively an image for each one of these micro lenses, and so you've got basically this sort of set of many, there's a whole array of images there, of which sort of some of these are sort of around this point have got sort of non-zero value there. And as you go above and below the plane of focus, this pattern basically changes. So below the plane of focus, basically you get sort of divergence across many pixels of the sensor. Above the plane of focus, you then get sort of converging and sort of less spread out. So you can use this information basically to tell you things about the sort of so to come. Well, I'll come back to that in a second. So what it lets you do in principle is instantaneous 3D imaging. So we've got here it's a 50 micron brain slice labeled with GFP. This is what you might see in a wide field picture. If you look at the unprocessed light field, you get something like this. So this is all of your sort of rays here. And then if we look in, sort of zoom in at this sort of box here, you see something like this. And then what you can do, so that's what it might look like unprocessed. You can then do various sort of processing steps, the simplest of which is this sort of synthetic refocus algorithm, where you can get back effectively to what the wide field would look like. Yep.
It seems from your data here that you have purposefully selected a high density of microlenses, and that you basically still have somehow high XY resolution, but not so much in Z. If you look at the synthetic refocus, everything shows up in plane at the same time. Or am I missing something? Why did you pick that trade-off? Do you need wide field of view maybe?
I mean, I think it is a trade. I mean, so there is a trade-off with. I mean, you obviously lose some spatial resolution with this, and I think we've got a slide in a minute, I think, which tells us something about that. So what are you asking about exactly? That.
So you get to pick.
Yeah.
You can trade off, for instance, have more microlenses per pixel or less microlenses per pixel. And here, based on the density of neurons that you have, you can resolve them in the XY domain quite well. So you could afford to have bigger microlenses, lose some XY resolution, and you would get perhaps better Z selection. But it doesn't seem like that's the option you picked. So I'm wondering.
I'm not sure whether that was. I mean, I think they're trying to get this going, and this was just this is not necessarily the tissue they want to use it for in the end. It's just a sort of a first principle application, and yes, you can trade that off. Yeah, exactly. And they're aware of that.
It's also only a 50 micron slice, so it's not a large volume of tissue that's being imaged at this.
Yeah, no, that's right. Where are we? Okay, and so basically you can synthetically refocus back to the plane of interest. Now, from my understanding, if that's all you were doing, you probably, you may as well not really use light field, right? But we'll see in a minute there are better things one can do with it. And so what you're doing is you're sacrificing lateral resolution for angular information. So before you had this sort of resolution, the sort of classic 0.6 lambda over numerical aperture, and instead, as we were just discussing, it sort of depends on the micro lens pitch, basically, and the magnification. So you've probably lost out a little bit compared to the wide field, and you can see an example of that there. Okay. So in this case, it's a 125 micron micro lens pitch with lateral resolution of 5 micron, and I mean, really, what Amanda is wanting to do is voltage imaging. So I think this is probably partly the selection there. Okay, so it's got some catches. So it does tick these boxes, but there are some catches. In particular, you've lost lateral resolution, and it actually is very computationally heavy as well. We'll sort of see that in a minute. Now, then this sort of goes on to the computational, so the image reconstruction techniques. So I mentioned synthetic refocusing. So in this case, what you do is reconstruct a single image at each plane, which represents the corresponding wide field image. So this is fast, but basically you don't have the optical sectioning, so you don't really win so much with this. But what you're doing, as I've understood it, is, so if you were to basically just average, so basically average the pixels under each micro lens, you would, that corresponds to this case on the left here. and it will. So, but what you do with the synthetic refocusing is sort of shifting. Effectively, you're sort of shifting the ray. So you've got access to the underlying sort of pixels, so you can sort of summate in a shifted way. You probably know more about them.
Yeah,
I do,
if
yeah,
you want
yeah,
very simply,
yeah.
you're getting your position and angle of your rays by adding this microlens.
Yeah.
So the data you collect
Yeah.
from your camera tells you about the position of rays, the angle, so
Yep.
you can basically just ray trace. That's
Yeah.
what the
Exactly.
focusing does.
Exactly. Yeah. So then, the more interesting algorithms are actually trying to get sort of 3D deconvolution from it. So you have this information. Can we make use of it to try and reconstruct a sort of a 3D picture of what's going on? So you're reconstructing the volume by deconvolving light field measurements with a 3D light field point spread function. Now, a classic algorithm for doing deconvolution iteratively is Richardson-Lucy algorithm. I mean, last year at this workshop we heard a lot about Richardson-Lucy. I don't know if we've heard it so far. Okay, I think this is where we were at before. So we've only probably got a few slides to go, so we've a lot of rigmarole for little. So we've basically done the light field acquisition, and then the job is to reconstruct the volume by deconvolution. And there are several algorithms that one can use. So basically, we've got a paper from the OSA proceedings last year. This is from Peter Quick, who was a PhD student with myself, collaborating with Amanda Faust, where we're basically sort of reconstructing the volume by deconvolution with a 3D light field point spread function. But in practice, what we often do is use iterative deconvolution methods, so the Richardson-Lucy algorithm, which we've talked about quite a bit, or these sort of class of algorithms, the paper from Broxton and Prevadal, etc. And these are more useful. You've basically, this allows us to have relatively good spatial and axial resolution. It's computationally slow. I think we'll see that in just a second. Time to analyze one recording of 150 frames using a particular computer spec. So if you just wanted to do that refocusing we talked about before, that takes over an hour. But sort of deconvolution for 21 iterations, and you may not need to go that far, but is around sort of 31 hours or so. And that's similar whether you use the Richardson-Lucy or the ISRA deconv algorithm. So, yep.
Is this coded up in Python or C or what is
It's in Python, in fact, yeah. Peter has, so we have an HPC system with many thousands of cores, and I know Peter did implement this stuff on there. So this is obviously not what these numbers are; they're just on one computer. But in practice, he is running it on this HPC system. So
more of a worst case scenario.
Yeah, well, I mean, this is kind of, I guess, just to illustrate that if you're sitting on your lab computer and you're expecting to do it, then it's not quite like that. You'll have to jump through some hoops if you want to do it quickly. So then, sort of finally, as I said, there are only a few slides to go. The two results slides that we've got are firstly, the first application was just in calcium imaging. So these are just slices bulk loaded with calcium indicator. I don't recall off the top of my head what the indicator was. It wasn't Oregon green. It was something else. So you can see this is sort of the wide field image. What you can get by using synthetic refocusing and the deconvolution algorithm. You can see the sort of the calcium transients. So sort of relative to so there's the wide field refocusing deconvolution. So these are the calcium transients you're seeing. If you look at the signal to noise ratio of these, then what you can see, so basically you're amplifying the signal, but you're getting sort of more noise as well. So it's in pure signal to noise ratio terms, the refocused one is actually slightly better. But if you want to be able to get things, individual neurons out of a 3D volume, you may, you're not going to necessarily be able to do that with the refocus method. So actually applying the deconvolution algorithms is your only bet there, and it's
One reason I go to
Yeah.
Multiphoton is not necessarily for greater signal to noise, it's
Yeah.
deconvolution, but source
It-
separation. So do
yeah, exactly. So sorry, you're saying did we do source separation or? Sorry, yeah.
Do you have metrics for that? What are the figures Aberrant used to say how well an algorithm can decouple signal from two neurons that are close together?
There is something on this in this first of the paper with the calcium imaging where Peter has actually done that. Yeah. So we've got a sort of a separability measure he's gone through and quantified. And I remember going through and doing some vaguely ROC-like analysis with him on this. We sort of worked something out. So it's in the paper. The details of that are in this paper. It's not that one, but it was one by Peter Quick. Anyway, sorry.
Why do you have two different SNR comparisons? Because refocusing works in the top one.
Yeah.
Better in the bottom one. So it's
Yeah.
wondering.
Okay, and I have to make apology because this is Carmel's slides. I'm kind of just trying to give a somewhat botched delivery.
Maybe for the top and bottom images.
Yeah, but that's not deep. Yeah, he doesn't explain that. Yeah, I mean, okay, these are obviously one difference is these are trains and these are individual ones. I'm not sure the answer to that off the top of my head. I'm sorry. Yeah. And then finally, the application was to voltage imaging, which is where we wanted to go. So so this is the reference here. This Frontiers in Cellular Neuroscience paper. What Peter Quick, the student, what we wanted to do with this was he was wanting to basically patch a single neuron, image, many cells both in the same plane and in different planes, and measure postsynaptic potentials and use it as a sort of a way of mapping, the synaptic weight matrix in effect from that neuron. So a sort of which you could also do by dual patching, but the idea is to try and scale this up. So that was his sort of motivation. We never quite got there during his PhD, but this was the kind of, the sort of reasoning behind development of this. And these were done with various of Thomas Knopfl's voltage indicators. This is the VSFP butterfly with sparse expression, which for these applications, at least it's the first thing to apply to. And so anyway, this is sort of the proof of principle, possibly first proof of principle of light field voltage imaging. I don't know if anyone else has done it yet. So if we can actually get this done, then we'll actually be first. We've collected a bit more data and are hopefully ready to send the paper in the next few weeks. So Peter is working on this now. But so this is basically the setup. You can see again this point with the deconvolution versus refocused. You're getting a bigger signal. You're also getting more noise. So the SNR is not necessarily better. Well, certainly not better. And they're able to pull out. These are just sort of three different neuronal processes. Yep.
The work of Nico, he did in the past tackle directly the problem of deconvolving.
Yep.
Have you thought about that as well? Meaning that, what can give you a good guide is which one of the two methods gives better separability in some sense?
Yeah, we haven't, as I said, we've looked at it a bit with the calcium. We haven't looked at it with the voltage indicator. But
these traces,
yeah.
they look a lot like calcium traces, actually. This must be a slow indicator.
So it's intentionally a slow indicator, a slow voltage indicator, and the reason for that, so we were aiming at PSPs rather than action potentials, and of course the thing about action potentials is they're very short, so there's not much time to collect photons, and so we kind of said actually we're better off aiming for you're intentionally using something with a slightly longer kinetics, slower kinetics. Yeah.
But that's also actually doubly let you use all the tools that you have for calcium imaging. Because the traces,
Many of, I would say, yeah, exactly. There may be some differences, but it may well be that you can reuse a lot. Yeah, yeah. Whereas I guess with your voltage indicating, your voltage approach, you're looking specifically at things which are aimed at that millisecond time resolution. Yeah.
But also, that's 500 milliseconds.
Yeah.
500 milliseconds, basically time. It doesn't buy you much using voltage indicator, if that's the. I'm looking at the
time bar there. So yeah, I mean these are.
At the end of the day, that looks.
These are I mean, but I think it's just because they're electrically. They would be electrically evoked. In that electrically evoked signals, no. So. Yeah, I mean, the time resolution of this guy is about 20 milliseconds, and we can sort of do this. They've done this at a few hundred hertz, so that's the resolution that we can get. So, yeah, the reasoning behind the time resolution again, like we got PSPs maybe sort of 20 milliseconds at most, so we need to be sort of certainly above 100 hertz. So a few hundred hertz is the sort of minimum you really need to get at this. So this was all aimed at a system for being able to map postsynaptic potentials, and
one spike, and then they get that one, or it's a train spike.
Yeah. So this is I think this is single. Yeah. Yeah. I have to ask her. Unfortunately, so she's given me some notes here, but they don't correspond to the things that people are asking about. Yeah. Yeah. Exactly. I can check that. She would obviously be able to deliver this much better, but I believe they were basically just electrically evoked, and a pair of pulses there that you can see. Yep. So that's just the finish of that second talk on Carmel's work. So this is. Okay, so she hasn't, of course, listed herself there because she was actually going to be delivering these, but so it's Carmel Howe, Amanda Faust being the PI of their group, and Peter Quick, a student, working. So my PhD student, so he's no longer a PhD student, he's graduated, he's now back as a postdoc with Amanda. And Pierluigi Dragotti, they're collaborating with, working on basically sort of more advanced sampling algorithms for light field, where you can basically take into account prior information. So there's a next stage of that they're working on at the moment with Pierluigi's group. So yeah, and there we are. Yeah.
So you presented this time that is. I'm assuming you are working on algorithm to make it faster, right?
Right at the moment, no,
so
because right at the moment, no, because they're focusing on the microscopy and data side of it. But I mean, we will be when that then becomes a factor again. So yeah, for sure, yeah. I mean, in the end, with basically depending on what comes out of this stuff, it may change the picture in terms of that, right? I mean, probably not for the better in terms of computation time, I suspect, but yeah. Well, definitely will.
This algorithm we developed is more about separating sources than it is about resolving sources.
Oh, but automatically will resolve sources.
It will.
I mean,
it
you
will.
will know what they are.
The one argument I would make would be that the ray tracing is part of the photon.
Yeah.
While the deconvolution basically may remove some of the information that you do care about.
For sure. Of course.
And so that would be a potential hybrid if you want to separate two sources.
Yeah, that's generally the thing with computational imaging, right? crank the handle on. You are going to throw away information, but
But
hopefully keep the stuff that you do care about.
I see
if it's
that
right,
becoming
yeah.
really handy
Yeah.
with faster
Yeah. Yeah.
voltage dies.
So yeah. Well, anyway, yeah, I think there are now a few groups that have got light field stuff going, and so I think it will be area of development for sure. Yeah. Okay.
Good.
Thanks. And